Okay, hello everyone. My name is Dawn Casey. I am the Director of Business Operations at Paragon Genomics, and I will be serving as the chairperson for today's event. On behalf of Cambridge Health Tech Institute's Breakout COVID-19 Global Web Symposia Series and Paragon Genomics, we'd like to welcome you to SARS-CoV-2 sequencing and high-throughput detection. So without further ado, I would like to introduce our speakers for today. We have three speakers. Our first is Lily Liu. Lily is the Head of Technical Support and Applications at Paragon Genomics. We also have Dr. Dien Bard. Dr. Dien Bard is the Director of the Clinical Microbiology and Virology Laboratories at Children's Hospital, Los Angeles. And lastly, we have Dr. Gai. Dr. Gai is the Director of Bioinformatics at the Center for Personalized Medicine at Children's Hospital, Los Angeles. So thank you in advance to our speakers for joining us today. Thank you to our attendees as well for joining us. So at this point, I'd like to pass it off to Lily Liu to go ahead and get us started.
Thank you so much, Dawn, for the introduction, and thank you everyone for joining us. In the first part of this webinar, I will introduce everyone to Paragon Genomics and its SARS-CoV-2 panel for next-gen sequencing. I will share some data and also how it can be adopted for high-throughput laboratory settings. Next, I will follow up with some introductions to our new products in the pipeline. That includes our efforts to support COVID-19 susceptibility research, influenza detection, and novel RNA virus research and detection. Without further ado, let's start off with an introduction to Paragon Genomics for those who might not yet be familiar. The company was established about 5 years ago and headquartered in Bio Bay of California. Our mission is to provide high-quality NGS library preparations and analysis solutions, specifically to advance precision medicine and research. Our products and technologies are patented and manufactured here in-house at our headquarters. Within the past few years, we have developed an extensive customer base all around the world with more than 400 custom targeted panels for various applications. The products we offer are based on our CleanPlex technology, which is the foundation of our SARS-CoV-2 panel. And what is this CleanPlex technology? It is a highly multiplex PCR-based targeted enrichment solution for NGS for confident detection of low-frequency variants with low sample input. To put that in layman's terms, we enable you to discover more with less. Our key features include low DNA input that actually allow confident genotyping even on the single cell level. And our chemistry also allows highly sensitive and specific detection that enables confident variant calling down to 1% allele frequency with minimum sample input. Our workflow is easy and fast, and it is the #1 feedback we receive from customers. The standard library prep takes about 3 hours with a 3-step single tube workflow. Our panels can be highly multiplexed, up to 20,000 targets per pool. Along with this high multiplexing capability, the chemistry also allows highly uniform amplification. So this ultimately translates to cost savings on sequencing. Our chemistry is compatible with various sample types and sequencing platforms to fit your application and research needs. Lastly, we pride ourselves in being able to offer fast custom panel turnaround, probably the fastest in industry. If you need a custom targeted panel, regardless of your application or the organism, as long as we have a reference genome, we can generate this custom panel and deliver the completed kit to you in as little as 4 weeks. Let's dive into the SARS-CoV-2 panel. The CleanPlex SARS-CoV panel was launched back in early March this year. Now, about 5 months later, we have shipped out hundreds of kits to labs all around the world. And it all started with the design. Our bioinformatics and R&D teams put extensive thought into designing this panel. As illustrated in the mathematical model shown in Figure A, first, we wanted high sensitivity to achieve the highest theoretical LOD. Naturally, this would depend on the number of fragments you're trying to detect. Then we have to balance the average amplicon length to ensure not only sequencing compatibility with various sequencers, but also sufficient length to allow high-quality primary designs. Additionally, the panel design was cross-referenced to minimize interactions with background RNA, such as those from human or other viruses, to obtain the high specificity. In the end, we generated a 343 amplicon panel split into two pools to span the entire viral genome of SARS-CoV-2. And this is what we offer today as the CleanPlex SARS-CoV-2 Research and Surveillance Panel. The panel is currently compatible on two major sequencing platforms, Illumina and MGI. Ion torrent compatibility is being confirmed, which will ultimately allow you the flexibility to sequence on whichever platform of your choice. To understand how Paragon Genomics Solutions fits into the bigger picture, let's go over a typical detection and sequencing workflow. The samples are collected from patients typically via swabs or saliva collection. Then the RNA or total nucleic acid is extracted. For detection purposes, on the lower half, the most widespread method for direct antigen detection is by RT-qPCR. The CT value is determined, and from there, the sample is determined to be either positive or negative. For this route, it then stops there. If the sample is carried through the CleanPlex NGS library preparation, the amplified product can similarly be used for detection purposes via electrophoresis visualization of a characteristic peak around 280 base pairs. But additionally, the product can then be sequenced to yield the full genetic information for additional phylogenetic and epidemiologic value for research or surveillance purposes. We have also partnered with Sophia Genetics to offer data analysis solutions for those of you who might need it. What does the library preparation entail, and how simple is it? Well, the workflow is quite simple. The workflow starts with the reverse transcription of the extracted RNA to cDNA, which is then carried into our standard CleanPlex workflow. And that consists of three main steps. The first being MPCR with target-specific primers, followed by a digestion step in the form of a short heated incubation. Then lastly, an indexing PCR step that barcodes and attaches the appropriate sequencing adapters of your choice. In this case, Illumina or MGI. Each of these steps are then followed by a magnetic bead purification. This entire protocol can be completed in less than 6 hours with about an hour of hands-on time, which really enables single-day turnaround for the entire sample processing, including RNA extraction. Given that the steps are purely master mixes, thermal cycling, and magnetic bead purification, the workflow can be easily integrated into most automated liquid handling platforms, which then allows higher volume processing and hand-free library prep. So hopefully the workflow seems easy enough, but how does the panel perform? Here we have some data with clinical samples. In the top left, we see a fragment analysis of a library generated with only 8 viral copies. This was actually prepared by a first-time customer and by the CHLA team. The operator was able to generate high-quality, clean libraries with their very first run, so it really attests to the fact that our workflow really is easy and robust. To the top right plot here, it shows the percentage of genome covered at 10, 50, and 100x. These samples were sequenced at 150,000 pair end reads. And on the bottom left, we also see consistently high mapping and on-target rates for these samples, which further showcases the specificity of the design and that little to no reads are actually being wasted. The CleanPlex technology, when compared to other similar technologies, is able to provide higher coverage with fewer total reads, also because of its uniform amplification, which I will share a little later on. On the bottom right, we were able to confirm the quality of amplification by performing variant calling on a control synthetic RNA sample from Twist with known sequence information. As shown in the table here, we were able to easily and confidently call the 4 expected mutations, 3 of which being SNPs and 1 deletion, with the expected 100% frequency. The strategic design combined with the robust CleanPlex technology really allows this panel to be used for many different applications. Anything from as simple as detection or confirmation of potential RT-qPCR false negative results to strain or haplotype confirmation to more research-based applications such as surveillance and tracking or intra-host mutation analysis. Given the high specificity and sensitivity of the assay, customers have also applied our system to environmental samples or samples from other hosts or investigational sample types, such as postmortem brain tissue. The SARS-CoV-2 panel is being run by labs around the world as we speak, and we have obtained a lot of great feedback, but we didn't stop there, because here at Paragon Genomics, we are overachievers. We understand that as the virus evolves, mutations do occur, and potentially it could occur at our priming regions that might later affect the binding efficiency and thus affect the overall coverage. Although there hasn't been any evidence or indication of this for the past 5 months, our team is already staying 1 step ahead of the virus by generating a set of degenerate primers to cover the observed polymorphic regions based on sequences that have been collected since the beginning of the pandemic. This is just us taking proactive measures to maintain the already high coverage that we're seeing to ensure the performance of the panel with time. Again, the team feels that it is important to plan ahead, and therefore we will be offering the Flex, which stands for flexible, version of the panel to those who are interested, such as those in the front lines of variant detection research, to give you that additional confidence to be able to capture these mutations right at the moment that they're emerging. Also, based on customer feedback, we've also included a human RNA primer pair to serve as a control to more confidently call negative samples. The positive control amplifies as a much larger amplicon at about 480 base pairs, which can be easily distinguished via fragment analysis. The cherry on top here is that the Flex panel also amplifies with slightly higher uniformity, as shown in the plot on the lower right. You can see from the amplification distribution versus the %GC plot on the bottom left, the panel shows very uniform amplification and no bias with GC content. Given the high performance, even at low read depth, and the ability for automation, this really opens the doors for high throughput and high multiplexing of samples for low cost sequencing. For applications that do not require full or deep sequencing, such as for detection, the assay can be reduced to a one-pool workflow, which effectively halves the total amplicon count, using just 500,000 pair and reads per sample for detection purposes. Even on an iSeq, you can sequence more than 300 samples per run, which calculates to probably less than $2 per sample in sequencing costs. For higher throughput applications that require more coverage, Paragon Genomics does provide various index offerings that, when coupled with NovaSeq's ability to run 8 lanes concurrently, it will allow sequencing of more than 3,000 samples at the same time. So this highly sensitive detection method with the ability to gain nucleic acid level information, we add, it really can add additional value to each confirmed case, such as helping with disease control efforts. The data presented in previous slides showed that CleanPlex method can offer high ratio of coverage and sensitivity to sample read depth requirements. This makes the CleanPlex panel stand out amongst other methods such as hybrid capture and metagenomics. That wraps up our SARS-CoV-2 portion of the presentation. Now I'd like to take the time to introduce a few new developments in the Paragon product pipeline. First, the team has designed a panel covering the entire coding sequence of ACE2 and TEMPEST2, which are two proteins involved in the internalization of the SARS-CoV-2 virus. Increased expression of these proteins have been linked to increased susceptibility to infections and also increased severity of the disease in those who do become infected. We tested a total of 11 cell lines to validate this panel. As you can see, the uniformity, mapping rate, target rates are superb. Everything pretty much at 100% or 99%, as high as it gets. We analyzed and confirmed the SNP genotype for some of the cell lines that were well characterized. From the table, you can see the heterozygous and homozygous calls with 100% concordance for these cell lines, which is as expected. There is still a lot to be learned about mutations in these two proteins and how it might affect its expression or affinity for SARS-CoV-2 virus. We hope that this panel can spark interest and facilitate additional research into how these mutations can affect individual susceptibility to not only SARS-CoV-2, but also to coronavirus in general, especially as we plan ahead for battling future infectious diseases. On the topic of future infectious diseases, the flu season is just around the corner. We can't help but to think how this year's flu season would impact COVID testing and vice versa. In response, the Paragon Genomics team is also developing an influenza detection panel specific to influenza A subtype H1N1, H3N2, and influenza B. We have selected unique regions in each of the 8 viral segments demonstrated by the gray sections in this figure for the purpose of identification and detection. We envision that in practice, when combined with the SARS-CoV-2 panel, it can offer more clarity for patients being tested because of symptoms that overlap for both diseases. We are actively validating this panel and hope to have it ready for beta testing before the flu season starts. Obviously, not all viruses are well studied, and therefore Paragon Genomics created a method for comprehensive RNA virus detection as well. The team is also actively validating this multiplex PCR-based metagenomics method, which is currently actually undergoing beta testing. It shares the same easy and simple workflow as our SARS-CoV-2 panel actually. Preliminary results using control material, in this case plasmids with N and S genes, shows very high and uniform coverage with only about 400,000 reads per sample. Hopefully you're picking up the theme here, and what we do at Paragon Genomics is to create technologies that focus on enabling high quality data with minimum sequencing or minimum sample input. Therefore, going back to our original model, we really enable you to discover more with less. The goal has been to make an impact by offering solutions for testing and research during the pandemic and with our CleanPlex technology. So to summarize, we have an expertly designed SARS-CoV-2 panel with highly sensitive and highly specific applications. Our CleanPlex workflow is simple and fast and allows for easy automation integration for high throughput environments. The panel produces highly uniform libraries with high on target and mapping rates that allows more cost efficient sequencing. The Flex panel of the Flex version of the panel was created proactively to maintain that high quality data for confident variant calling. We have new products in the pipeline, one being ACE2 and Tempest-2 panel for coronavirus susceptibility research. Another is for influenza detection to be coupled with SARS-CoV-2 panel in the upcoming flu season. And lastly, introduction to our multiplex PCR-based metagenomics method for novel RNA virus sequencing with low read requirements compared to a lot of other metagenomics methods out there. I like to leave everyone with a reference to our coronavirus application page at paragongenomics.com, where you will be able to find additional resources and data, as well as our bioRxiv preprint that was done in collaboration with the CHLA team. The publication is currently being considered, or the preprint is currently being considered for publication on Nature Scientific Reports. So with that, I'd like to pass it off to Dr. Dien Bard for the second half of this webinar, which will showcase her team's COVID research powered partially by Paragon Genomics technology.
Thank you, Lily. So we are about 8 months into the pandemic since it was first identified in December 2019 in Wuhan, China. So this slide here represents the data from this past Monday, where we were just shy of 20 million cases. And as of 9 a.m. when I looked this morning, we are at over 20.1 million cases with 737,394 global deaths. Specifically in the US, we have greater than 5.1 million cases and greater than 163,000 deaths. So as you can see from this global map, the US is really on fire with COVID-19 cases. And unfortunately, COVID-19 will be with us for likely some time. Oops, sorry. Okay. So with that, testing becomes extremely important to understand overall community rates, community transmission, and to try to mitigate the transmission, especially when paired with contact tracing and self-isolation. So testing can occur at different stages, ranging from the asymptomatic or pre-symptomatic period to the symptomatic phase, followed by finally the convalescent stage. So the ultimate goal of testing at any of these stages is to identify COVID-19 positive individuals and to then potentially control the transmission of these of SARS-CoV-2 from the positive individuals to others. This is something that we have done at CHLA since mid-March when we went live with testing. And so this would incorporate, patients, family members, employees that are tested as well. Testing at the convalescence phase for clearance was something that was more readily done in the past and has been replaced with more of a non-testing strategy based on the CDC's recommendation, as we are now more aware of individuals that can basically shed the virus for months on end even, and are not necessarily considered infectious. So now with a strict isolation timeframe from the time of symptom onset or test positivity, you can approach it with a non-testing strategy for clearance. So although severity and overall mortality of COVID-19 have been lower in children compared to adults, there's been still significant illnesses that have been reported in children. So in fact, one of the first cases of COVID-19 that we identified in our laboratory was on a 10-day-old male infant that was born at 39-week gestation. This individual presented to an outside hospital ED with increased nasal secretion and labored breathing, was found to be hypoxic with respiratory failure, and the patient was transferred to our PICU for higher level of care. The chest X-ray is shown here, and you can see this bilateral ground glass opacity with no focal consolidations. So it was suggestive of a viral etiology, potentially. We tested for SARS-CoV-2, which was positive from both the nasopharyngeal swab, the nary swab, and stool. And the patient was treated just with high-flow nasal cannula and eventually weaned off into room air. However, upon discharge, the patient was readmitted 5 days later with, again, increased work of breathing, was found to be still positive for SARS-CoV-2, which isn't surprising, but also for a secondary virus, so human metapneumovirus. The patient was stable, though, and was discharged without intervention the next day. So it's just an example of some of the cases that we've encountered since the beginning. The genomic epidemiology of emerging viruses has proven to be a very useful tool for outbreak investigation and for tracking of the viral evolution and spread. And the tool has really been used to further understand this particular virus, SARS-CoV-2. This figure here that I have inserted is just from a paper that was published in Science this year from the team at UCSF that investigated the genomic epidemiology of SARS-CoV-2 in Northern California from late January to mid-March. They basically looked at a metagenomics approach, sampling samples from 36 patients spanning 9 counties, as well as the Grand Princess cruise ship. So the phylogenetic analysis revealed the cryptic introduction of at least 7 different SARS-CoV-2 lineages into California, including the WA1 strain that was associated with the Washington State cluster. They noted a lack of predominant lineage and limited transmission among the communities, and the findings supported that contact tracing, social distancing, and travel restrictions really helped contain the spread of SARS-CoV-2 in California at that time, as well as other states. So I will now turn it over to Dr. Gai, who will discuss the extensive work done by his team in the development of a robust SARS-CoV-2 genomic and bioinformatic program in our Department of Pathology and Lab Medicine. Dr. Gai.
Thank you, Jen. So I'm part of the Center for Personalized Medicine at the Children's Hospital, Los Angeles. And as a center, we offer genomics-based diagnostic testing for constitutional disorders, pediatric cancers, and non-infectious diseases. So as actually Jen mentioned, Dr. Dien Bard, I just mentioned, the genomic epidemiology can be a very useful tool for controlling and fighting back against this pandemic. In fact, there was a joint report by the National Academy of Science, National Academy of Engineering, and the National Academy of Medicine recently pinpointing the importance of genomic epidemiology as a key component of any strategy to fight against this pandemic. So it's very important to know. And to us, and as a genomicist, I guess it comes natural. That probably was the only thing that we knew at the time how to do it to contribute to this war against this pandemic. So in doing so, what we did was, first of all, trying to develop a SARS-CoV-2 whole genome sequencing assay. So we evaluate a number of currently available research assays, including the AMPLISEQ SARS-CoV-2 research panel, also the Paragon Genomics CleanPlex panel that Nikki just explained to you, and also the Trace Bioscience SARS-CoV-2 research panel, which is a hybridization-based panel. In addition, and so our team, I'm actually director of bioinformatics for the center, so my team is a bunch of very talented bioinformaticians and developers, so that we develop a number of bioinformatic resources. Because nothing else, the lack of informatic resources at the time for analyzing the SARS-CoV-2 genome data. And with these tools and this assay, of course, and we can actually engage on a number of genomic epidemiology studies, trying to understand this disease pathophysiology of COVID-19. I'll actually talk about 2 of the epidemiology studies, one on the essentially publicly available SARS-CoV-2 sequences from the US, and another on the SARS-CoV-2 sequences collected from all across the globe. Then Jen will, in the end, will actually tell you about another epidemiology study of our own really just study the genomic sequences of our own pediatric patients. So start with actually the whole genome sequencing assays. This is actually a comparative study of a couple assays I'm showing you here. On the top, I'm showing the results from this torrent SARS-CoV-2 sequencing panel, and at the bottom, I'm showing the results of the Paragon CleanPlex SARS-CoV-2 research panel. These are the results on exactly the same 15 SARS-CoV-2 samples that we collected. These samples have viral loads ranging from 15 or 16 actually copies, estimate the 16 copies per microliter to about 500,000 copies per microliter. The way you look at the plots are actually, you look at the x-axis is depth of coverage, and then the y-axis is the fraction of the SARS-CoV-2 genome that's covered by at least a certain depth of coverage. The three horizontal lines actually, in fact, indicating the fraction of the genome covered at 95%, 90%, 80%. Then actually the three vertical dash lines indicate the depth of coverage at 50x, 100x, and 250x. Clearly, you can see that regardless of the initial viral load, for all 15 samples, we have obtained really very good whole genome coverage, close to 100%, in fact, whole genome coverage at even 50x and close to 100x as well. So that's actually far sufficient, of course, for the variant coding and the sequence analysis purpose. And the coverage is also quite uniform as well. If you look at the depth of coverage plotted for each of the genes encoded by this virus, ordered from the 5' end to the 3' end for these same 15 samples, showing this set of 15 plots. The red bars actually representing data from the on torrent, in fact, the Amplific panel, and the green bar representing data from the Paragon CleanPlex panel. You can see that the coverage indeed is quite uniform from the 5' end to 3' end for all the genes across SARS-CoV-2 genome, with exception, of course, for a couple of samples, say maybe in the middle. There are obviously a lot more sequences, a lot more coverage for the genes on the 3' end, and that may be actually for biological reason, maybe there's abundance of these subgenomic RNAs on the 3' end. So that's actually very good result. And I think there was actually a question asked by one of the attendees regarding how you actually differentiate maybe SARS-CoV-2 from other coronaviruses, or even SARS-CoV-1, that's actually the virus causing the original SARS disease. So this is actually in the advantage of using a sequencing-based assay like the Paragon Genomics CleanPlex assay, right? So after not only actually detecting the presence of amplicon, but after the sequencing, you can do the sequence analysis, which will give you the specificity you desire and give you the definitive answer as to how specific this detection is. In this case, we just actually align the sequences derived from these samples that actually showing two different viral genomes. Shown on the left is actually the alignment percentage of reads actually aligned to this original bad coronavirus. Of course, we know that the SARS-CoV-2 originated from this bad coronavirus. You see that there is indeed actually a significant percentage of the reads for each of the sample aligned to this bad bad actually coronavirus. And on the far right, and this actually alignment of percentage of the reads aligned to the SARS-CoV-2 sequences. Of course, you can see that they're higher than the essentially the percentage of reads aligned to the bad coronavirus. Showing in the middle are a number of alignment against a number of coronaviruses that are known to cause human diseases. You can see that barely actually any reads aligned to the 5 viruses that are known to cause human diseases. But on the second on the right, that's actually the alignment against the original SARS-CoV-1 genome, you can see there are still some reads aligned to this genome. Because of SARS-CoV-2 and SARS-CoV-1, they do share a significant sequence similarity, so that's expected. But again, just the sequence analysis really give you the confidence and the sensitivity and the specificity you desire for definitively diagnose certain infection. Now with the sequence, of course, you can do this sequence analysis and you can do the variant analysis. Because of accuracy of this sequencing assay, we can actually compare the similarity and the relatedness of the samples quite well. In this case, we just use a simple dissimilarity matrix to measure actually the number of different positions between two sequences. The one I color in red, these are the essentially sequences that are almost identical to each other. You can see that their difference is only 0. So they are really identical at every single position. And this is nice work done by Alex Rudolph, that he wrote a suite of tools that allow us to do this kind of sequence analysis and viral similarity analysis. I forgot to mention that the first set of results I just showed you, those are actually generated by another actually very talented bioinformatician in the group, Moise Bodawala, and he's really done a great job as well. Now, with actually the viral sequences and the variants detected, of course, you can not only look at the individual variants, but you can look at the variant profile, try to group the samples into different categories, like we're doing here, and this is in collaboration with Zhitong Liu and from Paragon Genomics in the process to try to optimizing the assay, right? You can see that in the 13 samples, clinical samples we sequence, they can largely be grouped or categorized into 3 different groups, group 1, 2, and 3. You can look at the variant profile, and they are largely similar to each other. What's interesting to me is that this is a work done in as early as March, I believe, when we were actually first validating the assay. We can already see some actually newly emerged variants that are different or absent in the reference sequence, the original virus identified in Wuhan, China, right? So you can see that there's a number of variants that are almost present in most, if not all, the samples, including the very famous D614G variant, the second variant shown on the right and the arrow. Okay, if you can see that this is actually A23403G variant. This is a variant in the spike protein is known to cause high transmissibility. And this is actually very interesting to see that how these variants may potentially lead to different pathogenicity and different infectivity and so on. So we're very happy to be part of course, the effort developing this assay, helping to develop this very robust assay that we like a lot. In fact, it's because of that we use this assay quite extensively for our genomic epidemiology studies, sequencing our own samples, which I'll tell you. As part of our effort trying to fight against this pandemic, we also develop a number of bioinformatic tools, and so including the SARS-CoV-2 genome app. This is a genome map. That's actually an application for annotating the SARS-CoV-2 variants. And also this resource that we call CHLA COVID-19 Analysis Research Database, or CARD in short. That's actually a suite of database of SARS-CoV-2 sequences collected not only by us, but also deposited by others in the public repositories. And also we built a suite of Phylogenetic tools that will allow us to do the phylogenetic analysis to measure similarity of all the sequences. This is nice work done by Li Shuangshen of the group. Both tools actually are available on the openly accessible on the web. And the first one, the SARS-CoV-2 genome map, was developed by Peter Chiska, a former postdoc of mine, and who's actually done a great job developing this tool for annotating SARS-CoV-2 variants. Now with this assay and the tools, we can actually do a number of things. And then there's CARD, and just briefly about the CARD, and there's something already useful, hopefully, to many people who are doing the genomic epidemiology type of study of SARS-CoV-2. We want to actually have a centralized genomic resource that contains all the full-length SARS-CoV-2 sequences, but also the reporting functions for both our own internal needs, but also for external needs for people's own research needs. So we have a large collection of full-length SARS-CoV-2 sequences, and we have a number of bioinformatic tools for variant calling and annotations. We also have a pre-computed phylogenetic trees, which will allow you to assess where each sequence of interest may actually origin from. Now, this is just a screenshot of the SARS-CoV, the CHLA COVID-19 analysis research database or card you can access, and it's openly accessible. This also, of course, if you're interested in the details about the kind of data we have in these data resources, the kind of tools that we have available for your own research needs, and please just go to this manuscript that preprint, in fact, we made available on our archive. Now, with this set of tools and the assay, in fact, we analyze a number of our own samples that just show you the utility. This is to show you the utility of such genomic epidemiology type of assay. This is a very early stage, and this is in March, so that there are only about 2,500 full-length SARS-CoV-2 sequences deposited in the database at that time already. And by doing the phylogenetic analysis, we can see that our samples colored in green, in fact, clustered sometimes together with sequences from other parts of the genome, other parts of the world or the nation, but sometimes they cluster by themselves. They're clearly indicating a local pattern, right? The pattern of a localized transmission, which is quite interesting. And similarly here, and you see that zoom in and look at another cluster, is that the number of CHLA sequences clustered by themselves, and together with actually maybe a couple of sequences from Minnesota. Looking at a bigger cluster, and it belongs to, and there are some sequences from New York, and have some sequence similarity with the sequences that we obtained from our own patients. It's quite interesting. You can see that the emergence of some localized transmission patterns, localized clusters, which I actually will come back to. Now, we also actually provide the informatic to allow you to not only look at the collection of SARS-CoV-2 sequences altogether as a cohort, but also looking at individual SARS-CoV-2 sequences of interest. In this case, and you can actually copy-paste just the sequence of SARS-CoV-2 sequences of interest here, and click annotate, and it will tell you a number of the variants detecting this sequence, and I'll tell you that its annotations and how much similarity it has with maybe publicly available SARS-CoV-2 sequences. So remember, I was telling you that we already actually realized that there seems to be a localized transmission pattern, and so we did this analysis, try to see if it's indeed true, not only actually at the local level, city level, but also at the state and the country level. What we did was actually collect a full genome sequence of about 6,000 published SARS-CoV-2 sequences collected in US strictly, and try to look at if there actually some pattern can be reviewed. So very quickly, and we actually find something very interesting. We found a total of 921 variants, and representing a total of 264 different haplotypes. Haplotypes being defined as a collection of variants, right? That works as a distinct signature of a given virus. And there are actually 4 very common variants, including this very famous D614G variant, 23403A2G variant, right, in the spike protein that leads to higher transmissibility. Altogether, we're talking about 921 variants, including not only the missense variants, but also some in-frame deletions, stop gain and loss mutations, which are quite interesting. We found a total of 77, in fact, US specific haplotypes, meaning that these haplotypes only present in US but not in any part of the country, but also some state-specific haplotypes, especially in Washington and California and Utah. These all point to the effectiveness of travel restrictions because of increasing number of state-specific haplotype in the states that implement these travel restrictions the earliest. And if you look at this, and you see that the US-specific haplotypes, in fact, somewhat clustered together or intermingled with haplotypes derived from isolates all across the globe. But at the same time, and they're actually distinctly different from some other clusters. meaning that there's a huge amount of diversity. I'm biased, I guess, and in believing that although the virus has a very slow mutation rate, in reality, and you have a large pool of mutations, some to say like for the virus to work with and to predispose this risk of developing emergence of some more transmissible or more pathogenic viral isolates. So that's just my personal fear, of course. And for this reason, I also look at over 50,000 reported full-length SARS-CoV-2 sequences from across the globe. We found a lot of so-called country-private or country-specific variant profile and haplotypes. Again, just to review, there's a huge amount of diversity existing in SARS-CoV-2 genome if you look at them as a population. So this is actually of concern to me. But coming back to the main message I want to actually share, everyone, is just say that in fact, genomic epidemiology is a very powerful tool, very useful tool for dealing with this pandemic. And I'm just glad that we have a chance to work with Paragon Genomics in developing and in utilizing a very powerful whole genome sequencing assay. So now I will actually hand it back to Dr. Dien Bard, and she'll tell you about another study that we did.
Thank you, Xiaoru. With all of that Xiaoru has described, it really does emphasize the fact that there is a need for detailed genomic epidemiology studies of SARS-CoV-2 across a wide range of patients. Being from a children's hospital, we are particularly interested in cases associated or caused in children where there really is a paucity of data. So the ability to really deploy genomic epidemiology approaches is particularly important as COVID-19 shifts from the current pandemic state to a more recurrent seasonal infection, as is what's speculated for the future. Large-scale genomic epidemiology studies of COVID-19 in children will also provide insight into how viral evolution and possibly host response can contribute to the severity of the disease. So with that, we really hope to capture pediatric-specific COVID-19 genome data across a wide range of geography, and are currently working on a multi-center study with a number of other pediatric institutions. What I wanted to present for the next few slides is just a small initial study that we conducted and submitted on the clinical features and biogenetic diversity of 35 COVID-19 patients that were identified at Children's Hospital LA. So during an 8-week period, so between mid-March to mid-May, we identified 35 pediatric patients with confirmed COVID-19. A median age was 12.5 years, and 13 of the 35 patients were admitted to the unit for further care. The remaining were outpatients at either clinics, pre-op screens that we do, or visited the ED, but were not deemed critical enough to be admitted. 2 of the 13 patients that were admitted directly to the ICU, and the overall median length of stay of the patients in the hospital was about 4 days. 40% of the patients that we did see had pre-existing conditions, with asthma being the most common feature. No patients reported travel history. However, 4 of the 35 did have direct contact with an individual that was COVID-19 positive. And no deaths, fortunately, was observed in our cohort. So while most reports suggest that SARS-CoV-2 can cause pretty asymptomatic to mild infection in children, over about more than half of our cases that we have seen in this 35 cohort, but also in a larger cohort that we're currently looking at, were found to be symptomatic. Of the patients that were symptomatic, the median time from onset of symptoms to positive SARS-CoV-2 result was 2 days. We had clinical data available for 20 of the patients that we looked at, and of that, 70% were symptomatic, including 2 that were pre-symptomatic when tested and then progressed to additional symptoms. The number one most common symptom was fever, followed by congestion, cough, and shortness of breath. There were other symptoms that were noted in small numbers of patients, such as wheezing, chest pain, even diarrhea, sore throat, and headache. 4 patients had X-rays conducted or CT scans, and 3 of the 4 patients that had chest X-rays performed showed opacities in the lungs. 5 of the patients required oxygen supplementation, and 3 were identified to have chronic conditions in addition to the COVID-19. And we only performed serology on 3 of the patients, and as per just standard of care of what the primary physician would have ordered, and all 3 were found to be positive within about up to a 10-day time frame or so from the time of positivity. We are currently expanding this, as I mentioned, to over 100 to 200 patients, just to really gather as much clinical data as we can get on COVID, on pediatric patients, just because it certainly is not as well known or well studied compared to the adults. There was also a direct correlation between a higher viral load and symptomatic patients, as well as young patients. So patients that were symptomatic had high viral load compared to asymptomatic patients. And patients that were younger, so less than 5 years of age, also had a higher viral load compared to older individuals. And this correlates with a recent study that was just published looking at viral loads in younger children compared to older kids, where they did also report a higher viral load in the younger kids. So this is a figure illustrating the single nucleotide variations of the SNPs and the evolutionary rate of SARS-CoV-2 genome that was obtained from the 35 pediatric patients. So in the first figure up top, it demonstrates a spread of the SNPs across the genome in the 35 isolates as compared to the Wuhan isolate. So the vertical black line on the genome indicates the presence of the SNP. And the upper panel, of course, is just the structure of the SARS-CoV-2 genome. So of the 35 sequences that were obtained, it reviewed about 97 unique SNPs and 7 insertions or deletions compared to the Wuhan isolate. So an average of about 8.9 unique variations per isolate. What was interesting, and what Xiaowu also mentioned earlier, is that we found a predominance of the D614G mutation in the S protein. So 94% of the isolates actually had that present. And as reported before, this mutation was shown to be rapidly fixed in isolates from Europe and North America, and has been postulated to be more transmissible compared to the other. We also looked at whether genomic diversity in the SARS-CoV genome can predict disease severity, but at least in this small cohort, comparison of viral genome did not identify any variations that could help to separate out the symptomatic versus asymptomatic patients. However, with a potentially larger cohort that we're working on, we may be able to identify certain key variants. The next figure to your left represents a summary of characteristics of SNPs in open reading frames and non-coding regions, as well as the predominance of a non-synonymous variation across different open reading frames, which correlates again with what's been found in the literature. The predominance of the non-synonymous variations across the different open reading frames, as I mentioned, has been previously documented and really highlights the evolution of SARS-CoV-2 during the course of the pandemic. And it'll be interesting to see as we get more samples in the coming months how this changes further. And the last figure to your right illustrates a time resolved evolutionary rate estimate using the 35 isolates. So the evolutionary rate calculated using metadata that we had for each isolate determined a 6.4 times 10 to the negative 4 substitution per site per year, which correlates with the mutation rate of about 6 times 10 to the minus 4 that's been reported in the literature. The inferred time to the most recent common ancestor based on the molecular clock analysis of these isolates was December 4th, 2019, which again is comparable to what's been reported in the next strain of December 6th, 2019. So it's just a nice check balance for our method as well as our analysis to be able to generate similar data. Okay. And then this is a a phylogenetic tree that demonstrates genetic relatedness between our 35 isolates against a number of other isolates that have been reported globally. So there are certain conclusions that can be drawn from this. One is that comparison of the 35 isolates against about almost 1,000 genomes from the US and globally shows that the CHLA isolates certainly cluster predominantly with others from the US, but you also see that it does have similarities from certain isolates from Europe and Australia as well. We had one that the arrow down below here is a sibling pair, and you can see that it does cluster together because they are found to be genomically indistinguishable. So overall, the diversity among the CHLA isolates points to multiple potential introductions of the virus into Southern California from across the US and globally as well. So in conclusion, we have incorporated a SARS-CoV-2 whole genome sequencing as part of our COVID-19 diagnostic testing algorithm. So every positive that we've identified by our viral PCR test is then reflexed to whole genome sequencing at the CPM. It is a powerful tool to really help us identify local transmission when paired with contact tracing and metadata. And we've been able to utilize this tool to investigate potential transmissions within the hospital or to rule out potential transmissions within the hospital, and that's been very useful. Sequencing of the viral genome has allowed for an increased understanding of the viral genetic diversity in circulating strains of SARS-CoV-2 in Southern California. And lastly, genomic epidemiology should really be considered an essential component of any pandemic response strategy. And this is just a list of individuals at our institution that we'd like to acknowledge, including our department chair, Dr. Alex Judkins, the division chief of CPM, Dr. Jackie Beagle, and then the three in bold here that has worked immensely hard, as well as the entire team at CPM, as well as the clinical virology and microbiology laboratory. It's been a long, hard 6 months or so of just nonstop work for us, but it's also been very rewarding to be able to generate a lot of this data. And with that, I'm going to hand it over to the moderator.
Great. Thank you, Dr. Dien Bard, and also Dr. Gai and Lily Liu for all of the great information. We certainly appreciate everyone's efforts in all of the research and hard work that has been done over the last several months. At this point, what I'd like to do is to move us into our Q&A session. We do have time for a couple of questions, and we have been asked a few questions. So what I'll do is start with a question for you, Lily, I believe. The question is, what is the impact of pre-analytical variability on viral samples to test the test sensitivity, specifically heat time, type of VTM, especially for saliva samples, and what VTMs do we recommend for optimal results?
Our panel is specifically designed with the shorter amplicon length so that it is better at amplifying products that might have some degradation in the during transport, prior to actually performing the library prep itself. So we have considered that in our design itself by forming very short amplicons so that even if the sample itself is degraded, chopped up into little pieces, you're still able to amplify that product. Regarding other specific impact, that's hard to say. Paragon Genomics hasn't specifically done that. Maybe it's something that CHLA, Dr. Dien Bard or Dr. Gai may be able to comment on regarding what they have seen between different types of samples versus how it impacts the final results. Regarding the VTM, I believe that's the medium in which to carry saliva samples. At the same time, we're not the expert in that. So unfortunately, I don't have the best information to answer that question.
So I guess I can add to that with regards to the different sample types, at least from the PCR standpoint, because I think that's where it's probably plays is most impactful when it comes to detection of the virus or not. From the sequencing side, and it's my understanding that regardless of the sample, as long as you have good, nucleic acid, that it really shouldn't affect sequencing. We've included some non-respiratory samples in our sequencing runs before, including stool, and it actually has not been an issue. From the detection standpoint, we are working with saliva right now. We actually just submitted our data to the FDA for EUA so that it could be used as a clinical test. And so far, the performance of saliva compared to nasopharyngeal swabs has been equivalent, and at least in our hands, and for first-time positive individuals. For stool, we do know that it can shed a lot longer in the stool compared to other sites. However, working with even just the extraction process from stool is definitely much more difficult, and there's a lot more inhibitory substances. So it may be a little hit and miss whether you get proper good nucleic acid to then proceed on with amplification and sequencing.
Okay, great. We do have a couple of questions for you, Dr. Dien Bard, and also for Dr. Gai. So there was a question in terms of have there been any studies done showing the correlation between different polymorphisms of ACE2 and TEMP-PSS2 and viral loads?
That's a good question, and I unfortunately am not too aware. I haven't really looked into the host factors too much. Dr. Gai, I'm not sure if you are aware of that, the differences between ACE2 and viral load.
Just, I guess this is actually a project that we wanted to pursue, but have not had the time and energy because of limited bandwidth, right? And it is something we want to look at not only from the viral genetic perspective, but also from the host genetic perspective, maybe to look for the interplay between the host genetic factors and the viral genetic polymorphism to see how this actually could maybe conservatively lead to higher transmissibility or lower transmissibility. So it is a very interesting question, but we have not had the time to look at.
And there was also a question on one of the slides, I believe that you presented, Dr. Dien Bard, where you listed the 35 CHLA patients. There was a question in terms of if any of those 35 patients were admitted and how long before they tested negative.
13 of the 35 patients were admitted, and we didn't necessarily, I'm trying to think how far back. So typically they were negative, usually within about, I would have to say probably 10 days or so. However, we have identified patients, not necessarily within that 35 cohort, but patients and employees that have been positive for, even up to 60 days or so, completely asymptomatic, but just continuous viral shedding. And I think that's also one of the reasons why the CDC has changed their policy now to adopt a more non-testing strategy approach, where it's based on the timeline from the symptom onset of I believe 10 to 14 days, rather than depending on 2 negative test results.
Okay, great. There is one more question for you here. So this is one of the first questions on the analysis side. So is the expression limitations in alternative splicing by spliceosome with epigenome regulation is due to specific protein isoform in the protein pool that do not participate in the feedback control of protein expression? So the question is the expression limitations in alternative splicing by spliceosome with epigenome regulation is due to specific protein isoform in the protein pool that do not participate in the feedback control of protein expression?
I don't what has something to do with viral genetics or the host genetics, honestly, and I am lost.
Question, what we can do is follow up with the person directly, since we will have all these questions along with email addresses, so we can follow up directly regarding that question. So I think at this point, this really does conclude today's webinar symposium. I do want to thank our speakers once again for all of their time, all of their great efforts in helping us to better understand SARS-CoV-2 and advancing research so that we can, as a community, better combat the disease and certainly treat patients. Thank you to Cambridge Health Tech Institute for hosting us today. And as a reminder, this webinar is being recorded. We will send you an email directly from Paragon Genomics that will include the link to this webinar. So please feel free to share that with any colleagues that were not able to attend. So with that, I'd just like to say I hope everyone enjoys the rest of their day, and thank you for attending.